All right, so I'm going to leave this up for another minute while I talk, and then I'm going to have to take it down so I can put my slides up. If you're still having problems, come up and see me after class. Again, a lot of times it's just something you have to figure out on your own. You have to go in. Sometimes you got to change your password because you changed the password at some point and you forgot what it was because you did it 2 years ago when you had a different textbook or whatever. But I will help you if I can. Today is the last day that I will do a sign-in sheet. So if you still can't get this to work today, you need to figure it out over the weekend. Even if that means calling McGraw-Hill tonight. I know it's hard to call people, but you got to call them tonight if you have to, if you can't get it to work. Because a lot of times it's nothing that I can do about it. If it's not working, it's something beyond my control. But sometimes it is something I can fix. So you can come check with me. Before I take this down, does anybody have any questions about anything we talked about last time? About any of the policies or anything about the class in general? All right. Okay, so we're going to pick up, I think this is where we left off. We talked about microbes harming humans last time. Now we're going to kind of go back and we're going to talk a little bit about microbes in history. And the history of microbiology, how microbiology was developed as a science, how long it took, things like that. We're going to talk about some of the major discoveries in microbiology. Keep in mind, like I said last time, in general, you're not going to have to remember dates. Dates are not important. I'm never going to ask you what date something happened. However, as we go through, you do have to remember the names. So we're going to talk about several scientists and what they discovered. You do have to remember their names and what their contribution was. Okay, what their ideas were. We're going to talk about some experiments. You do have to know the experiments and what they proved, but you don't have to remember the dates. Okay? All right, so before we can kind of talk about the discovery of microbiology, I want to go way back and talk about what the primary ideals were way before microbiology was a science. Because we're going to talk about this in a second, but microbiology has only really been a science for less than 200 years. So it was the late 1800s, like the 1890s, when microbiology was really discovered. Before then, and I know that's a really weird concept, because you have grown up in a world, and I did too, and my parents did too, where viruses and bacteria are real things. You know, we may not understand all of it. If you've gone through your whole life without taking any kind of science class or anything like that, you may not know the specifics of viruses and bacteria, but you know what they are conceptually. You know that they exist and that they make you sick sometimes, right? But what did the world think before all of that was true? What did people think was making them sick? What did they think was happening before they knew about these tiny little organisms that they couldn't even begin to see, which is a crazy thing to think about. As far back as there are written records, there is evidence of people with infectious diseases existing. So obviously people were aware of infectious diseases because they had them. People got sick. But there was a lot of talk, and there are records of people with diseases being quarantined. So kind of in this early disease management. Specifically, a disease that used to be called, I say this name because it's what a lot of people recognize, but it's really the not PC term for it anymore, and nobody uses it, so you're not really supposed to use it either. But it's a disease that used to be called leprosy. Most people are familiar with that term because it's talked about a lot in the Bible. People with leprosy are talked about in the Bible a lot. Now it's called Hansen's disease, so I will talk about it as Hansen's disease from now on. That's the nicer term. Leprosy is not a nice term anymore. So Hansen's disease, as far back as there are written records, even in the Bible and well before that, in ancient Egypt, in ancient China, and all of these things, there are records of people being quarantined, meaning if they started showing these symptoms, people pushed them out of society and they were not allowed to live with other people anymore. Okay, they used to actually have something called Again, this is not a nice term anymore, but they used to have something called leper colonies, which is basically where anybody who had these symptoms was shoved into a colony and they all had to live together without any contact with the outside world. I believe that the island off of the coast of Louisiana, where I think it's Tabasco Island now, where there's like a little jungle anywhere you can go, that used to be a leper colony. So they would send people over there that started showing, and we'll talk about Hansen's disease later. Hansen's disease has very visible symptoms that a lot of people found distasteful, I guess. It can cause disfigurement and it can look kind of scary. But as far back as there are written records, there is record of quarantine, which gives an idea that even though they did not understand what was happening, they didn't understand that Hansen's disease was being caused by a bacteria, because it is, they understood that one person could make another person have it. Even though they didn't understand that it was a bacteria moving from person to person, they understood that something was happening to make the people near them get sick, right? And that makes sense. Even if you think about having like the flu, one person in your house gets it, everybody in your house gets it, right? The people that you're near are also going to get it. So it's kind of makes sense just based on things that you can see with your own eyes. Okay, so early disease management, a lot of diseases in early ancient times were thought to be acts of the gods. In ancient Greece, it was the gods. In, ancient Western cultures, it was just God. But whatever it is, it was thought to be an act of a greater power. Not something that was contagious or not something that, you caught from somewhere. It was something that you did, right? Which is obviously not what happens anymore, but that was the thinking at the time. But again, because they did quarantine, that's not how you spell quarantine, is it? I just realized I spelled that wrong. Quarantine, sorry. I'm already tired today, apparently. Quarantine. Because they quarantined people, there was this idea that being near somebody who was sick could also make you sick. Right? This is an early idea that diseases could be passed from person to person. In ancient Greece, the prevailing theory was something called the miasma theory. So the miasma theory, which was developed by Hippocrates, the miasma theory is the theory that disease was caused by something called miasma. And miasma was thought to be, miasma in Latin literally means bad air. So they thought diseases were caused by bad air, bad smelling, foul air that could be trapped in your house and make everybody sick. And it was thought to come from excessive amounts of dust or from rotting things. So if anything died nearby and things smelled, not keeping clean, if your body smelled bad or whatever else, there was this production of miasma. And the miasma would make you sick. So for a lot of people, disease prevention in ancient Greece had a lot to do with airing out your house. That if you aired out your house, you'd get the miasma out. Now, the funny thing is this actually isn't terrible advice, right? It's not going to completely prevent things, but a lot of diseases are airborne, and a lot of diseases are caught from dead, rotting things. And if you are in a house with something that is dead and rotting, the chances of you getting a disease are actually pretty high. So airing your house out, and this still holds true today. If somebody in your house has the flu, if you open up all the windows and you keep airflow in your house, it's less likely to spread. Now, that's not going to completely prevent it, but again, this isn't horrific advice. They stumbled onto something that actually kind of works. But again, they did not understand that what was in this bad air was not just bad smelling things, but viruses and bacteria. But that was kind of the idea in ancient Greece, was this miasma theory. And a lot of disease prevention focused on airing things out and preventing being around bad air, keeping things clean. If your house was clean and smelled nice, you were less likely to get sick, which again is not terrible advice. If your house is hygienic and things are kept clean, you are less likely to get sick. And the miasma was thought to basically, it was bad air, and it was thought of as like a poison. And it was thought that it would poison the body. And ancient Romans developed something in around 600 BCE, so before the Common Era. So they built something called the Cloaca Maxima. So this is actually directly translated to the greatest sewer. So I want you to think a little bit about how long ago this was. This was almost 3,000 years ago. This was 600 BCE. In ancient Rome, they built this huge sewer system. And it's marked, you can actually go to Rome still and see this. It is held up that long. It was an engineering marvel for its time. But they brought in all of these rocks and stones, and they built this huge sewer system. And this is a map of ancient Rome. And that big blue line is where the Cloaca Maxima goes. And it would basically carry out sewage. So it would carry away dirty water, and it would bring in fresh, clean water. And again, the fact that they were able to build this is one of the aqueducts from the Great Sewer, and that's what it looks like today. So you can still go to Rome and see these sewer tunnels, which is pretty crazy. And then this picture down here is a public latrine. So that's where people would go to empty their chamber pots or go to the bathroom. So they were public bathrooms. Nobody had private bathrooms at the time, but that is where people would go to dump all of their sewage, and it would get carried away by the Great Sewer into a nearby river, which caused problems for a lot of other people, but Rome was clean. So all of their sewage and human waste was carried away. And this Cloaca Maxima, they did not know it at the time, but the Cloaca Maxima was a huge part of the success of Roman society. It's not the only thing, right? The Roman Empire it was successful for a lot of reasons. But one of the reasons that it was successful as an empire was because of this sewer. And the reason was because in other parts of the world, like in England at the exact same time, or China at the exact same time, anywhere else, they were literally, especially in England, if you see accounts of England at this time, does anybody know what people did with their human sewage? They literally threw it in the street. It was very common. You had a bucket next to your bed, you went to the bathroom in it, and you woke up in the morning and you dumped it out onto the street, on top of anybody else who might be walking around. Their streets were literally filled with feces from every type of creature that was living in the city. And so disease, as you can probably imagine, was rampant. And it was very common, especially in European cities at the time, for there to be plagues every couple hundred years. And when plagues happen and half or 3/4 of the population is wiped out, your society basically has to start over every couple of hundred years. In Rome and in Greece later, but in ancient Rome, because they had this sewer, they didn't die out every 200 years. Their society was able to keep growing. Their knowledge was able to keep growing, and they were able to continuously get bigger. So a big part of the success of the Roman Empire and why they were able to get as far as they did was because they built a way to deal with their poop. Okay? They had a great sewer. And again, at the time, they didn't understand why this worked. They didn't understand about foodborne pathogens, and they didn't understand that feces has tons of bacterial illnesses that get passed from person to person. But they noticed that when they drank fresh water, they didn't die as often. Okay, and then our last one that we'll talk about kind of for ancient thinking is somebody called Thucydides. And Thucydides was, I believe he lived in Greece. Thucydides was a Grecian physician. who lived around 410 BCE. And he noted, kind of importantly, that in about 430 to 410 BCE, he made the important observation, that people who had gotten sick, so there was a very large plague that wiped through Athens around this time, and it killed almost half of the population. But what he noticed that was if people got this plague and they didn't die, a lot of people died, but if they got this plague and they didn't die, they survived, that they never got that plague again. That they only got it one time, and then they could be around all these people that were sick and it wouldn't ever matter. They never got the plague again. So he made the really important discovery about the Athenian plague. Again, that people who got sick once and survived never got sick again, which is an early idea of immunity. And we're going to talk about immunity a lot in this class because we're going to talk about vaccines a lot. And so immunity is why vaccines work. It's why you typically will only get one disease one time. Now, I know a lot of us have had the flu more than once and things like that. Those are not actually the same virus. So you've gotten the flu more than once because your flu virus mutates every year. But for things that don't mutate, things like chicken pox, I know none of you had chicken pox, probably, because most of you got vaccinated. But I'm old enough that I'm older than the chicken pox vaccine. So I got chicken pox when I was 3 years old. I could hang out in a room full of toddlers with chicken pox, and I'm never going to get chicken pox again because my body already did it. Did it one time, I'm never going to get sick again. Okay? And it's a really important idea, immunity, which we'll talk a lot about in this class. But he was the first one to kind of make that connection that if you get sick with this thing one time, it doesn't matter how many other people you're around that have that illness, you will never get it again. All right, so kind of the beginning of microbiology and this idea of organisms being alive that are smaller than we can see with our own two eyes was around 1859. So again, a little less than like 200 years ago. Around 1859. And that scientist who was like the first one to really make strides in the world of microbiology was a scientist called Louis Pasteur. And what Louis Pasteur did was he discovered something called biogenesis, which I'll explain in just a second. So before 1859, before Louis Pasteur, the major theory or the major idea that existed was something called spontaneous generation, also known as abiogenesis. And abiogenesis, or spontaneous generation, is the idea that life spontaneously generates, or it can. Or that life creates itself. That living organisms can pop into existence out of nowhere. Spontaneous generation. Now, I know that that's a weird thing to think now, but at the time, think about things that if you didn't know microscopic organisms existed, if you looked at them with just your eyes, the things that you could observe with your own eyes might appear that things are popping out of nowhere. So some examples of this are maggots appearing on decaying things, right? Maggots appear out of nowhere. Or a nicer thing to think about, have you ever had it rain at your house and there's like a big puddle in your front yard and the next day you come back and there are a bunch of tadpoles in there? It happens at my house all the time. We have dips in the grass. We get these huge puddles when it rains and there's always thousands of tadpoles 2 days later. If I didn't know any better, I could see, it's like this is not connected to another water source. this water wasn't here 2 days ago, and now there's a bunch of living things swimming around in it. How else did they get there if they didn't pop into existence? Another one are things like weevils. So weevils are bugs that live in grains. And weevils can kind of appear, like if you've never had weevils at your house, consider yourself lucky. It's no fun to open a bag of flour and have a bunch of bugs pour out. But in things like grain stores, corn stores, wheat stores, things like that, sometimes weevils just pop up. Now we know that tadpoles are the result of frogs coming in at night and laying their eggs in those puddles, and then the eggs hatching and tadpoles coming out. Now we know that maggots are the result of flies coming in on something that's dead and rotting and laying eggs and hatching, and then those become maggots. We know that weevils happen because eggs are in those grain stores, and then the eggs hatch and the bugs appear. Even things like mushrooms popping up anywhere, in grass, on wood, in your house, mold showing up in your house, things like that. Now we know that funguses are spread through microscopic spores, but at the time, they didn't know any of that stuff. So they thought that it was possible for things to just pop into existence. Abiogenesis, or spontaneous generation. Now, Louis Pasteur disproved that theory in 1859. He disproved spontaneous generation or abiogenesis. Those are the same thing. And he did that, and it was basically the very beginning of microbiology, because in disproving spontaneous generation, what he was actually proving is that there is life so small that we can't see it with our own two eyes. And the way that he was able to do that was a very simple but very effective experiment. So what Louis Pasteur did was he developed these very special flasks. And this is what they look like. They were called swan neck flasks. And these flasks were specially designed by Louis Pasteur. He actually blew the glass himself for these. He designed them himself. And what they are is they're a round piece at the bottom, and it usually has some kind of broth in there. Okay, so a broth is something that would grow life normally. If it was exposed to the air, it would grow stuff. It's got enough nutrients that things would grow in it. It's like leaving a container of chicken broth out on the counter. It's gonna grow stuff in a few days, okay? So, but he created these swan-necked flasks, and he would put broth in the bottom of them, and then he didn't seal the top, but he left the top open, but in this swan neck shape, okay? So the only opening of this flask is this teeny tiny opening at the bottom of this very curved neck. Okay? And then what he would do is he would heat these flasks up until the broth was boiling. So anything that was in that broth ahead of time would be dead, right? It sterilized the broth and meant that there was no living cells in there. Vigorous heat is applied to produce a broth free of live cells, meaning that it's sterile. And what he noticed was with these swan-necked flasks, if the neck was intact, anything in the air, so the air was capable of coming in, right? Air doesn't have any weight. Oxygen, gases, they don't have any weight. So oxygen could come in and go through the flask, right? Because oxygen can travel through any surface. Oxygen is going to travel, gases are going to travel, no matter the shape. However, bacteria or dust or viruses or anything that is a physical, anything that has mass, a physical particle, is not going to be able to change direction, right? Dust does not go down and then go back up. So things like dust and bacteria and viruses or anything else would go into the flask, they'd fall in because of gravity, and then gravity would stop them right there. Okay, anything that was a physical particle, any living thing, any dust or anything like that would get trapped at the base of that swan-necked flask. They wouldn't be able to go past the curve and go back up against gravity, right? Again, dust can't go down and then back up. It just settles, okay? So what he noticed was when he did this, when he heated up these flasks and he left them intact, he could leave this broth out at room temperature in the open he had ones that went on for years, and not a single thing would grow in there. Nothing. They remained clear for their entire existence, as long as the neck stayed intact. Okay? So nothing would ever grow in there. However, when he broke the neck off of a flask, what that means is that the oxygen can drop right in because of gravity. But now the bacteria, the dust, the viruses can also drop right into the flask because now the top is open. So the bacteria, the dust, the viruses, they drop into this broth, and within 24 hours, the broth is cloudy, it smells bad, there are clearly things, it changes colors, there are clearly things growing in there. Okay? And so what this proved was the theory of biogenesis. Not abiogenesis, biogenesis. And what biogenesis is the theory that life, all forms of life, are all forms of life are born from other forms of life. Nothing is created out of nothing. Okay, there is nothing that just pops into existence, nothing alive. Anything that is considered alive becomes that way because it came from something else that was alive, right? And with humans and animals and things like that, that's easy to see. Humans birth their offspring. An entire human exiting another human is a pretty obvious thing. Even stuff like flowers and plants. You have one flowering plant, it creates seeds that you can usually see, and then you have more plants. But with things like bacteria and funguses and weevils and tadpoles, sometimes it's not as easy to observe that happening, but it does. Nothing in this world that is alive comes from nothing. Everything is born from something else that is living. Now, the way that happens depends on the type of organism. Bacteria don't give birth, right? We'll talk about how they reproduce, but bacteria don't give birth. And so this was kind of the very beginning of microbiology as a science. This proved, by proving biogenesis, Louis Pasteur proved that there were organisms that were alive that were too small for us to see. And they were getting caught in those swan-necked flasks, but they were there. There were things that were alive that we couldn't see with our own two eyes. And so that was the beginning of microbiology. This was one of the most important discoveries in microbiology as a science. And again, I know that looking at this now, it seems really dumb. Of course, if bacteria can't get in there, the broth's not gonna grow. But again, you grew up in a world where bacteria existed. Think about how freaking mind-boggling this must have been in 1859. To go from not knowing that there were these teeny tiny little things that existed on every surface to then knowing that. How crazy that must have been. Now, I know it seems dumb because you all know that bacteria exist. But back then, this was a very revolutionary idea, that there were two organisms that were too small and they were covering every surface of the world that we lived in. Okay, so biogenesis is the currently accepted theory of the world, that everything living comes from something else that is living. It has not been disproven. There's lots of evidence that goes for it. It's the currently accepted scientific theory about how life is created, biogenesis. Okay, So another really, really important discovery in microbiology, as you can probably imagine, is the microscope. And the microscope is not an organism. It's a tool or an instrument that allows us to see microorganisms. So it allows us to see things that are too small for us to see without aid, I guess. And the very, very first microscope was discovered in 1665. Now, that is a big difference, right? This is like 200 years earlier than Louis Pasteur. It just goes to show you that sometimes scientists aren't believed for a long time after they discover things. But Robert Hooke was the first person to develop a microscope, and he developed the first one in 1665. And this microscope was strong enough to view eukaryotic cells and multicellular organisms. So it was able to look at large things and see them on a smaller scale. For example, his very famous picture that he developed in 1665 is this one right here. This was actually published in one of the first scientific magazines. Now we have hundreds of them. Back in 1665, there weren't that many. But this is his famous picture that was published in a scientific magazine in 1665. And that is a picture of cork bark from a tree. So he took a piece of cork bark and he put it underneath his microscope that looked like this. So this is his first microscope. And he was able to see the details of the structure of the bark. So instead of just seeing a piece of bark, he was able to see the details and what made up the structure. And the word cell, we use the word cell. Things are made up of cells. Most of us have heard of the word cell before. The word cell to describe a unit of life was actually coined from this picture right here, because he looked at that bark and he said, oh, it's made up of individual little cells, because he could see the individual little compartments within the bark. And he was the first one that said, it looks like a bunch of little cells of things. And he came up with the word cell. Okay, because he could see all of those individual little pieces that were making up this bigger organism. And so he was kind of the first one to develop or come up with this idea that large organisms like humans and animals are made up of lots of tiny things called cells. Okay. And then the second microscope developed was in 1674, so not that much longer, by Antony von Leeuwenhoek. And Antony von Leeuwenhoek was he developed the first microscope strong enough to view true microscopic organisms. So unicellular and microscopic organisms. And he is most famous for he would look at things and see these funny little organisms floating around in whatever he looked at. Specifically, he looked at water. So he would take drops of water, and that was his microscope down there. And he would put drops of water on that microscope, and he could see little organisms swimming through the water. He didn't know what they were. He didn't know what they were called, but he saw these little things swimming through the water. And he famously called them wee little beasties. He was a Scottish scientist, and called them wee little beasties. But they were just little tiny things that were swimming through the water. He also frequently looked at the plaque scraped off people's teeth. Plaque in your mouth is covered in bacteria. It's basically made up entirely of bacteria. So he would scrape the bacteria off people's teeth and look at them under his microscope, and he'd see them swimming around in there. And so he was one of the first people to view bacteria. Now, next we come to a stage in microbiology where medical microbiology was developed. And there's a lot of other things happening at the time, but again, this class, the focus is mainly medicine and human health. So we kind of focus on that in this class. So as you can probably imagine, soon after the just mind-boggling discovery that our world is covered in these tiny little organisms that we can't see, pretty soon after that, people started thinking, wow, that probably means that they're all over our bodies too, and that they're in everything that we touch, and they probably get in our mouths, and they probably get in our noses, and they're probably making us sick. And so soon after discovering that these things existed, they started to realize that, hey, this probably has an effect on human health. And so medical microbiology was kind of born from this time period, soon after Louis Pasteur developed his swan-necked flasks. Now, sterilization, as in general, sterilization is the idea of eliminating all living things, including spores. Now, I know you don't know what a spore is yet, or you don't know it in the context of bacteria, or you probably don't. We're going to talk about spores later, but I'm just putting this here for now, that sterilization is the idea of eliminating everything that's alive from a surface or from an environment. And you've probably used the word sterile in your lifetime. A lot of times when we use it like colloquially, just kind of in our everyday life, we're not actually using it properly. So a lot of, like, one of the things that people talk about is like sterilizing baby bottles. You're not actually sterilizing a baby bottle. You're removing most of the bacteria, but you're not removing everything. But sterilization is the idea of removing everything living from an environment or a surface. And this, again, is an idea that we use pretty frequently today. We talk about sterilizing food products, or we talk about sterilizing makeup. Makeup comes sterile. A lot of things come sterile. If you ever had to have surgery, you had surgery in a sterile room, and they cleaned up your body, and they made your body as sterile as possible before they cut it open. So sterilization is something that we still use today. But at the time, they were just discovering that sterilization was important. Now, aseptic techniques are a series of processes to achieve sterilization. Okay. So aseptic technique is a series of processes, a series of techniques, a series of things that you can do to achieve and maintain sterilization. And again, aseptic technique may be a word that you're familiar with. If you're taking the lab, or if you take the lab at any point for this class, aseptic technique is a huge part of that. You have to learn how to handle bacteria aseptically, meaning that you don't sterilize things. If you ever are a nurse or a healthcare worker or a doctor or anything else, you will have to learn how to do things aseptically. If you are cleaning up somebody's wound, you don't want the bacteria from your hands getting slapped into their open wound, right? You have to learn to put gloves on and clean the area and not touch other things with your clean gloves, right? If anybody has ever watched any medical show in existence, you've watched doctors scrub in, right? They're performing aseptic techniques to prevent bacterial growth. Okay? This is why when you go get shots, they clean the area first and they put on gloves. It's because we don't want to inject bacteria straight into your bloodstream. That's a bad idea. Okay? But again, a lot of these things that we use today, hand washing, the fact that we have surgical suites, the fact that it's normal to, clean the area with alcohol before you get a shot, the fact that when we get tattoos or piercings or anything else, everything is sterilized ahead of time. All of these hygiene practices are baked into our existence. And most of us are pretty familiar with these. right? You brush your teeth twice a day. You wash your hands after you go to the bathroom, or you should, right? These ideas are built into what we do. It's been a part of our lives since before we were alive. However, at the time, those ideas were ludicrous, just completely insane, and they didn't exist. So the first doctors, or the first people who introduced these ideas, were two doctors. The first one was Ignaz Semmelweis, and the second one was Joseph Lister. Now, Ignaz Semmelweis was a physician. I believe he was Hungarian. He was Hungarian. And he was practicing medicine around 1845. And I think he made one of the kind of coolest discoveries. I think it's an interesting story because it was just something that he observed in his life. And he took that and extrapolated it and created procedures that we still use to the day. Okay, so what Ignaz Semmelweis noticed, he was can you go get paper towels? Sorry. So Ignaz Semmelweis was a Hungarian physician who had sorry, I lost my train of thought. He had a doctor's practice, and he did all kinds of, at the time, physicians were not specific, right? Doctors at the time did everything. They performed surgery, they delivered babies, they did autopsies, they did everything. And he was a doctor in 1845. But at the time, it was also really common. It was kind of at a time when some women or some people giving birth went to a doctor to have a baby, and some people went to a midwife. Some people didn't have a doctor at all, but there were midwives who were specialized practitioners, and their only job was to deliver babies. They were special baby doctors, and their only job was to deliver babies. And they would cover many towns, and they would deliver all the babies in all of the towns. Okay? So what he noticed in his doctor's practice was that when he delivered a baby in his own practice, if he delivered babies, the baby's chance of death was incredibly high. So if a mother went to a doctor to have her baby delivered, the chance of her getting a postpartum infection, they didn't call it an infection at the time, but the chance of her getting a postpartum infection and her getting sick and probably dying, and the baby getting sick and probably dying, were incredibly high. Upwards of 50 to 60%. So 50 to 60% of the people who delivered babies with doctors got sick, and almost all of them died. Okay, those are big numbers. But when he looked at people who went to midwives, so the mothers who went to midwives to deliver their babies, the infection and death rate was less than 10%. That's a big difference. And he looked at that and he said, why? Why is that? It's not about the procedure, because the babies were born fine. It was later, it was after they were born. And he couldn't figure out, it wasn't something he was doing in the procedure, he was doing bad medicine or anything like that, and he couldn't figure it out why. But around that time was when Louis Pasteur came up with abiogenesis, and Joseph Lister was also around that time, and some other scientists that were really important. And he thought, hey, I wonder if it's this weird stuff that everybody is saying exists that we can't see. And what he also realized was, as a doctor at the time, he was not just delivering babies. What was he doing? He was going to see sick people. He was going to see sick people. He was going to autopsy dead bodies. He was going into people's houses where they had deadly infections. And he was walking straight from a dead person's house where he did an autopsy to a woman giving birth and delivering her baby. Okay? And this was all happening in 1845, where gloves certainly didn't exist. Right? And he was like, I wonder if I am passing something, I am picking up something from this autopsy and putting it in somebody else's body. Because giving birth is a very vulnerable place to be, microbiologically speaking. Okay? Things are very open. Things are coming out of areas. There's a lot of places for things to get back in. Okay? And so what he did was he developed the very revolutionary technique at the time of washing his hands. And when he washed his hands between patients, all that took, they developed lye soap, and he washed his hands in between patients. And when he washed his hands, going from one patient to another, his postpartum death rates dropped to that of midwives. So I think that's a pretty cool story, because he looked at something and was like, why is that happening? And the midwives are people that only deliver babies. So they weren't going to autopsies. They weren't seeing people with the flu. They weren't seeing sick people. They were just delivering babies. And when he did this one thing, he washed his hands with soap from one patient to the next, he saved dozens and dozens and dozens of people's lives. And obviously, hand washing is still something we do. And it's one of the most important and effective techniques in controlling bacterial infection, even to this day. And it was all developed because one doctor saw something and figured out a way to change it. I think it's pretty cool. Now, Joseph Lister so he introduced hand washing. And Joseph Lister was around the same time. And what he did was he used the ideas of Ignaz Semmelweis, who kind of came before him, and Louis Pasteur. And he noticed that it was very frequent for people to die after having any kind of surgery, right? You cut open somebody's body, the chances of bacteria getting in there is pretty high. And so what he did was he started the procedure of cleaning surgical surfaces with chemicals and things like that. He wasn't able to sterilize things at the time, but he used very strong chemicals to wash everything that was going to be used in a surgery. And then he also used, so this is a depiction of one of his surgery suites. And what that mist is that's actually a mist of something called chlorohexidine, which is not a substance that you have to remember, but it is basically the same thing that's in Lysol. So chlorohexidine is an early version of Lysol. And so what he would do is he had somebody in surgery that would basically mist Lysol over the whole open area. Now, getting Lysol in your body is not a great plan, but it also prevented bacteria from growing. And so he was able to decrease his death rates post-surgery by a significant amount by introducing these like surgical cleaning techniques. Okay, so he made everybody that was in a surgery wash their hands. They all had to push their hair back or have shaved heads, things like that, which are, again, things we still use today. Anybody in a surgery has to wear a surgical cap. Anybody going into surgery has to scrub themselves down. They have to wear clean, right? They're all wearing like aprons and towels. Now we wear scrub jackets, I can't think of the word, but scrub drapes and things like that. But all of the procedures that occur in a surgical room today exist because Joseph Lister did it first. And his name is actually what Listerine is named after, because the first version of Listerine had chlorohexidine in it, like he used in his surgical suites. Obviously, Listerine does not have Lysol in it now, but at the time when it was first created, it did. Now, kind of another important part of medical microbiology is this idea of germ theory. And germ theory, again, is something that you should be pretty familiar with, even if you don't know the name. But again, at the time, it was ludicrous. It was ridiculous. Diseases came from bad air, or the gods, or a curse from a witch, who knows? Diseases came from places that we couldn't explain. But the germ theory of disease, the first one that kind of talked about that was Louis Pasteur. And when Louis Pasteur figured out that there were these tiny little bacteria, tiny little things growing in everything, he very quickly discovered that those tiny little things were what was making us sick. So the germ theory of disease is the idea that diseases are caused by microorganisms. Oops. Microorganisms. So we kind of already talked about Louis Pasteur and biogenesis. Louis Pasteur was also arguably one of the most important discoveries in all of human health is he named a procedure after himself that you're probably all familiar with, pasteurization. So after he discovered that germs, or these things floating around in all of the substances that we couldn't see were what was making us sick, he started working towards preventing that from happening. And one of the things that he did was he developed pasteurization, which is something that we still do to this day, almost in exactly the same way he did it, all the way back then. So pasteurization was discovered in 1864, and it's really hard to overestimate how important pasteurization is to the overall survival of human society. Because milk and milk products and apple cider and beer and things like that have tons and tons and tons of bacteria in them. Cow milk is absolutely chock full of bacteria that will make you very, very ill. And pasteurization, and a lot of people, I think it's kind of crazy, and it's one of the things I like to talk about, is that I saw something, and who knows how reliable this is? This is not scientific evidence, but I saw something on TikTok, like everybody else, about a woman who was doing research, and she went around to like 1,000 people, and she asked them what they thought pasteurization was. And like 70% of them had no idea what pasteurization is. And that's not great. So I'm here to tell you what pasteurization is. Pasteurization, the very complicated process, is heating milk up to just below boiling and keeping it there for 15 seconds and then letting it cool down. That is pasteurization. Pasteurization does not involve chemicals. Pasteurization does not involve actually boiling anything. Pasteurization does not require the addition of anything else. It is heating something up to just below boiling, because if you boil it, then the taste is weird, right? If you've ever boiled milk and then drink it, it sucks. So you have to heat it up to just below boiling so it doesn't taste weird, and then you cool it down. And most of the disease-causing bacteria is gonna die just by heating it up. And because it's that simple, again, pasteurization is still done almost exactly the same way as Louis Pasteur did it back in 1865. Okay, just heating up the milk and cooling it down. But that's simple, and I think it's really good proof that sometimes the simplest thing is the best thing. Because again, I cannot overstress how critical pasteurization has been to overall human survival. Millions of people used to die from foodborne illnesses from milk every single year. Pasteurization has made it so that babies, human babies, can have formula. Okay? The existence of formula has saved countless babies, right? Because before that, if their mother didn't produce milk, or their mother died, or whatever else, the babies died. They couldn't feed them. After pasteurization was created, you could safely give them goat's milk, and then after that, formula was able to be produced. So there's the countless ways that pasteurization has saved arguably trillions of people's lives at this point. And if you want to drink raw milk, that's up to you. I will not be drinking raw milk, but I think it's important for people to realize, I think, again, a lot of these misconceptions about things that get thrown around on the internet and wanting things to be natural in the way that they're supposed to be. And I think that a lot of people do think that pasteurization is like adding a bunch of chemicals, adding nitrites, adding anything else, and it's not. I actually saw something one time on TikTok that from a person that drinks raw milk that said, oh, well, I drink raw milk. I don't want it to be pasteurized, but before I drink it, to make it safe, I just heat it up on my stove. Babe, you're pasteurizing it. That's what it is. All right, so I think it's just important to talk about what things actually are rather than some of the stuff that gets thrown around a lot by people who don't know. All right, and then Robert Koch was another scientist, and we'll talk more about him later, but Robert Koch was instrumental to figuring out which bacteria caused which diseases. So he was able to figure out the specific bacteria that caused anthrax and the specific bacteria that caused tuberculosis and lots of other bacteria. And so he was critical in the idea of this one bacteria causes this specific thing. So he overall was just really important to the idea of germ theory. Okay, so we're going to start in on this section. We probably won't finish it, but that's okay. So that's kind of all we're going to talk about when it comes to the history of microbiology. And again, I just think it's really important for you all to realize we live in a world where microbes have always existed. My mom lived in an area where microbes always existed. Her mom probably did. I'm trying to remember when my great-grandmother was born. But before that, no, I take that back. My great-grandmother was not born before that. My great-grandmother was born in like 1860. So she would have been born, I know I'm old, she would have been born right around the time that this was happening. So can you imagine being alive in 1860 and not having any idea that any of this was true? And then in your lifetime, the discovery of microbiology and bacteria and vaccines and medicines and pasteurization, it's wild to think about. Okay, so now that we've kind of covered the history on how some of this was discovered and how microbiology became a science, sorry, I have a lecture right before this one. 2 in a row is hard. We are going to talk about the kind of basics of microbiology, but just kind of science in general. So we're going to talk about the macromolecules. We're going to talk about a few other things, but we're going to be talking about the things that make up living cells. So what does something have to be in order to be considered alive? What are the things that all cells have to have? How are those things making us alive? Right? What are the rules? Okay. And these rules, most of them apply all the way. We share a surprising amount of DNA and processes and particles and all kinds of stuff with single-celled bacteria. There's a lot of things that you can find in a bacteria that you will find exactly the same thing inside of a human being. There's a lot of it. Okay, so we're going to talk about a lot of the things that they all share, and then we'll probably spend the next section talking about what makes them different. But this is the macromolecules. There are 4 macromolecules, and these are molecules that make up most components within any type of cell, whether that be a bacteria or a human. And macro, just as a prefix, remember again your prefixes, macro, what does macro mean? I think I heard it, big. Macro just means big. It's the opposite of micro. Micro means small. Macro means big. Macro molecules. So these are big molecules. Okay. And our 4 big molecules we're going to be talking about are, and you should recognize the names of all of these, carbohydrates, also known as sugars. Lipids. Does anybody know what lipids are also known as? Fats. Nucleic acids. Does anybody know what else that's called? That's kind of a tough one. The NA, it's DNA. So our DNA or our genetic material. And then protein, which is honestly just called proteins. But carbohydrates, lipids, DNA or nucleic acids, and proteins. Now, 3 of those 4 are main components in our diet. And we're going to talk about that as it kind of applies to nutrition and diet and things like that. But sugars, fats, and protein are what we have to eat to keep us alive, right? If you're not eating sugar, fats, and proteins, you will waste away. You have to eat them in order to survive. And that's because your cells are made up of those things. If you don't have those things, you can't make cells, and you will cease to be alive. Okay, so these 4 macromolecules are the basis of everything that a cell is including its physical shape and physical parts of the cell, what it's physically made up of, and it is responsible for everything that a cell does. Everything that a cell is and does is done or created by these 4 macromolecules. Okay? And they are called macromolecules. Again, macro meaning big. Another name for a macromolecule is something called a polymer. Poly, does anybody know what a poly means as a prefix? It means a lot or many. I think I heard you, many. A group of, a large amount of, right? It just means many. Or big, so a polymer. And something that all of these polymers or macromolecules share, one characteristic, is that they are all made up of smaller building blocks. Okay, and so they are made up of monomers. Mono meaning one. So the way that this works is that these polymers are made up of lots and lots and lots of individual building blocks called monomers, all hooked together. I kind of think of them like a Lego tower. A Lego tower is a bunch of individual Lego bricks, right? You have 100 Lego bricks, and you stack them all on top of each other, and they become a tower. And I'll use that analogy a lot, but in that analogy, the Lego brick is the monomer. It's the individual piece. Mono meaning one. It is the individual piece. And the tower that you create when you stack them all on top of each other is the polymer. It's the many pieces. So it's all of the pieces together, many. Okay, and we are going to talk about all of these polymers, and we are going to talk about the monomers that they are made of. So carbohydrates are made up of, carbohydrate is a polymer, the monomer of a carbohydrate is a monosaccharide. Lipids or fats are made up of fatty acids. And again, we're going to talk about each of these in turn. But that's kind of the overview. All right, so carbohydrates, our first macromolecule, are carbohydrates. Now, carbohydrates, again, are a polymer, also known as a macromolecule. Okay, all of those things mean the same thing. Okay, they're also called sugars. Okay, and then you'll see that the polymer also has another name down here. It's also called a polysaccharide. I don't know why everything has to have 5 names. It just does. I'm sorry about it, but it's true. Okay? So in carbohydrates, the monomer of the carbohydrates is usually a monosaccharide. So that was the easiest one to remember because the monomer is a monosaccharide. They both say mono. Monomers is a monosaccharide. And so these individual sugars, this is a monosaccharide right here. It is one sugar molecule by itself. Mono, one sugar. And monomers, or monosaccharides, their purpose within most organisms is typically for energy or food. Okay? Specifically, currently usable and short-term energy. Now that's going to make more sense in just a second when we talk about long-term energy, but this is short-term energy. Energy that your body will use to do whatever it needs to do. Everything that a cell does, from reproduction to, in a human body, having a thought requires energy, right? Breathing requires energy. Moving requires energy. Sleeping requires energy. Every single thing your body does requires energy. building the other macromolecules requires energy. And all of that energy comes from monosaccharides. Okay? Some examples of these are glucose, lactose, and sucrose. Those should all be pretty familiar names. Glucose is our primary energy food source as humans. Bacteria, a lot of them use glucose. Some of them use lactose instead. Lactose, where do you find lactose as a sugar? Milk products. Sucrose can be in fruits and other things. Glucose, lactose, and sucrose. Those are our big ones. Okay. Now, you can hook two sugar molecules together. So look at this picture over here. One sugar molecule looks like that. It's just like one little individual blue thing. And we're not going to get into the specifics of structure and things like that. So just don't worry about it. I'm trying to keep it as general as possible. But if that is what one sugar molecule looks like, you can also have something called a disaccharide. Now, di usually means two. Okay? Di means two, so that's two sugar molecules. So a disaccharide is just two of those monomers hooked together. Okay, so you have one monomer here and one monomer there. And they are hooked together by a bond, which is that black line right there. They're bonded together. They're a disaccharide. But when you hook together a bunch of sugar molecules, what you get is many sugars, a polysaccharide. So monosaccharide is one sugar, polysaccharide is many sugars, usually defined as 10 or more, but lots of sugars all hooked together. So the polymer is called a polysaccharide. Again, this one's easy. The monomer is a monosaccharide. The polymer is a polysaccharide. The other ones aren't that easy, but this one's easy. Now, polymers are, if anybody has ever crystallized sugar or melted sugar and like made something out of it, or did it on accident, you burnt sugar in a pan or something like that, you realize that sugars, especially if they're in the right configuration, can be really hard, physically hard. They are a hard substance. If you've ever melted sugar to a bottom of a pan, it's impossible to get off. It's physically hard. And so these polysaccharides, one of the things that they do is they are structural components. So they are physical components of cells that provide them structure. And we'll talk a lot about polysaccharides as we go through individual types of cells. And then another thing that polymers can do, polysaccharides, is that they work as energy storage. Or another way of saying this is long-term energy. Okay? So what I mean by that is that in a human body, something that happens, we're going to be talking about glycogen. Okay, so that's our next one. In a human body, think about what I just said, that everything that my human body does requires energy. Well, as I'm standing up here and I'm talking and expending a lot of energy, or you're out exercising and expending lots of energy, you're doing whatever it is, existing, expending energy, do I have to constantly eat? Am I constantly eating more energy? No. We as humans eat every couple of hours, right? We don't have to constantly be putting more food into our mouths to fuel what we're doing. So what our body does, so I ate my breakfast a couple hours ago. Right after I ate my breakfast, I consumed some amount of glucose. Most food has some type of carbohydrate in it. So I consumed a certain amount of glucose or lactose. I had cottage cheese, so I guess there was lactose in it. And my body used what it needed at the time. It was like, you need this amount of energy right now. And that is what the usable short-term energy is. It's the energy that your body needs right this second. But then what my body does with the extra energy that I consumed is it stores it. It actually turns it into, it gathers up all those extra glucose molecules and links them all together and turns them into a polymer or a polysaccharide called glycogen. And that human glycogen gets stored in my liver. It gets put in the little pantry that is my liver, and it sits in my liver until I need it. So right now, I haven't eaten in 4 hours. You can probably hear it, some people in the front. I'm hungry now. I haven't eaten in 4 hours. I don't have any food currently going into my body, but I still need to power the things that I am doing. I'm standing up here waving my arms at all of you. So what my body is now doing is it's taking that glycogen and turning it back into food sources. So that glycogen is a way of saving that sugar. It gathers them all up and sticks it in one spot and saves it for later. Okay, it's saving the leftovers, putting it in a Tupperware in your liver and saving it for later. So that's what glycogen is. Starch is very, very, very similar. It's a very similar thing to glycogen, but starch is what plants use. So it's what plants use to store their sugar molecules. So a potato is basically pure starch. When you are eating a potato, you are eating that plant's starch storage. So you have just stolen all of the energy that poor little potato plant meticulously saved up. No, I'm just kidding. But that's what they do. They store their energy as vegetables, as carrots, as turnips, as potatoes, and they're all full of starch, as corn. It's a starch. It's a storage form of that energy. And that's why when you eat it, there's lots of carbohydrates in them. Starch is a carbohydrate. Okay? We'll talk about chitin and cellulose